Got a few RNAs, MTR we don't play. mRNA carries info to the DNA in the ribosome. We bring tRNA to help correct the amino acids, and it's essential for protein synthesis. It helps to bind specific amino acids. RNA polymerase, synthesis 5 to 3. Switch it up, it reads DNA to 3 to 5. Two different sides, yeah that's right. Got a message from RNA, now description. Tell you in a minute. Whole DNA strand needs to be unwinded. Everything on the DNA is copied, but it ain't destroyed, it stays there. It's not killed, it's just duplicated by polymerase. Where is this? It's on the nucleus. Prokaryotes in the cytoplasm, but stop. We're just on eukaryotes.
Transcription involves RNA polymerase using a single template of DNA strand directing inclusion of bases of the brand new RNA. Initiation, elongation, terminations of prior translation. Ribosome, of course, reads the sequence of groups of 3 bases. To assemble the protein, yeah, that's crazy. Elongation, making polypeptide chain longer. Initiation is a lot of things. 1 is the promoter of a gene. 2 where RNA synthesis begins its routine. A binding site for RNA polymerase decides where transcription takes place. Yeah, DNA. Yeah, RNA.
I'm gonna switch it up a little more this time. I'm gonna show you how MTR really shines. 3 processes with them splicing and dicing, then at the cap tail that's icy. Last it exits from the nucleus, that's likely. tRNA matching appropriate codons in mRNA. rRNA inside ribosome helps to attach mRNA. A couple of the things you should know, bro. There's also alternative splicing. Excision of introns is enticing. Splicing and retention of exons, which is cool, can result in different molecules. RNA polymerase doesn't copy the DNA into strand that's complementary into mRNA during transcription, then the RNA wouldn't be existing, and the DNA can't leave the nucleus of the cell, which is ridiculous. Genetic code won't reach the ribosomes, where amino acids they could code to produce proteins. But here's something else you might not know. Nucleotides on mRNA are triplets called codons. I know that's a lot, but listen to me. There's one more run through is repeat. Transcription involves RNA polymerase using a single template of a DNA strand, direct inclusion of bases of new RNA. Initiation and elongation, terminations of prior translation. Ribosome reads sequences of groups of 3 bases to assemble the protein that's crazy. Elongation making polypeptide chain longer. Initiation is a lot of things. It's a promoter of a gene, where RNA synthesis begins its routine, a binding site for RNA polymerase, decides where transcription takes place. Yeah, DNA, yeah, RNA. Now let's see some bloopers, woo!
Got a few RNAs, MTR we don't play. mRNA carries.
Initiation, elongation, terminations of prior translation. Got my hizzy and my glizzies. Nope.
It's a lot, but listen to me. There's one more run through is repeat. Transcription involves RNA. I'm gonna show you how MTR. RNA matching appropriate codons MNR. Yep. I'm gonna show you RNA inside ribosome helps to attach mRNA. A couple of things you should know, bro. There's also alternative splicing.